This is Dr. Mitch Robinson, and this video is on glycogen metabolism, and it accompanies handout number 10 for cellular and molecular medicine. In this video, we'll examine the pathways for glycogen synthesis and breakdown and their regulation, and we'll describe disorders of glycogen metabolism. Glycogen is a readily mobilized storage form of glucose. As we've seen, it's a very large branch polymer with alpha-1,4 glucose residues with alpha-1,6 branches about every 8 to 10 residues. Glycogen can be broken down to yield glucose molecules when energy is needed. Most cells contain a small amount of glycogen as a reserve fuel for ATP production, but in the body there are significant deposits of glycogen in the liver and skeletal muscle. In liver, the primary function is to maintain glycemia and keep the brain supplied with glucose. In muscle, the function is to maintain a supply of glucose for energy production for contraction. In our big picture of metabolism, we can see that glucose 6-phosphate is the branch point for glycogen metabolism. Let's begin by looking at the pathway for glycogen degradation. Glucose molecules are removed from the non-reducing ends of the glycogen chains by the enzyme glycogen phosphorylase. The residues are removed by insertion of a phosphorus molecule, not water, so this is phosphorylasis rather than hydrolysis. The product is glucose 1-phosphate. Glucose 1-phosphate can be converted to glucose 6-phosphate by phosphoglucomutase. Glycogen phosphorylase can remove glucose residues from glycogen chains up to 4 residues from a branch. At that point, a glucan-transferase enzyme moves 3 residues from the branch to the main chain. This leaves 1 residue connected by an alpha-1,6 bond, and this residue is removed by alpha-1,6 glucosidase. This is also known as the debranching enzyme. So now that the branch is removed, the glycogen phosphorylase can again resume removing residues from the non-reducing end. So now we have glucose 6-phosphate produced from glycogen breakdown. In the liver, glucose 6-phosphatase can remove the phosphate and glucose can be transported out of the cell. In liver, glycogen provides glucose to the bloodstream. But in muscle, glycogen does not leave the tissue and is used within the cell for energy production. So muscle does not have glucose 6-phosphatase. So remember that the enzyme glucose 6-phosphatase is found only in liver and kidney, but not other tissues. And recall that glucose 6-phosphatase is found in the endoplasmic reticulum of the cell. This removes it from glycolysis. If the enzyme were present in the cytosol, it would prevent any glucose 6-phosphate from entering glycolysis. Okay, so now let's examine the pathway for glucose synthesis. Glucose residues are added to the non-reducing ends of glycogen polymer chains using glucose in the activated form, UDP glucose. The enzyme that makes UDP glucose is UDP glucose pyrophosphorylase. UTP is used and pyrophosphate is produced in that reaction. The enzyme that adds glucose to the polymer is glycogen synthetase. We also have a branching enzyme that takes 6 or 7 residues and moves them to make an alpha-1,6 branch. There's a protein called glycogenin that serves as a primer for glycogen synthesis. This is where the glycogen molecule gets started. This enzyme has the ability to add glucose residues to part of the protein chain. Okay, so now let's look at the regulation of glycogen synthesis and degradation. The 2 important enzymes in glycogen synthesis and degradation are glycogen phosphorylase and glycogen synthetase. Glycogen metabolism is under very tight control in the liver. The main function, again, is to maintain glycemia. Glycogen phosphorylase and glycogen synthetase are regulated by phosphorylation, dephosphorylation. The 2 enzymes are reciprocally regulated. That is, when one is active, the other is inactive. The phosphorylated form of the synthetase is inactive and the phosphorylated form of the phosphatase is active. A diagram on page 7 is meant to show how this works in the cell. The A form is the active form of the enzyme, and the B form is the designation for the inactive form. Notice that the phosphorylated form of the synthetase is B, or inactive, and the phosphorylated form of the phosphatase is A, or active. So when both enzymes get phosphorylated, synthetase is inactivated and the phosphorylase is activated. Glycogen is broken down and glucose is released. If both enzymes are dephosphorylated, the synthetase is active and the phosphorylase is inactivated. Glycogen is being synthesized. Primary control is through extrinsic regulation by the blood hormones insulin and glucagon. Insulin causes dephosphorylation of both enzymes. That increases the activity of glycogen synthase and decreases the activity of phosphorylase. Glycogen synthesis is activated and glucose is stored. An increase in glucagon in the blood causes phosphorylation of both enzymes. This increases the activity of glycogen phosphorylase. Glycogen is broken down and glucose is released. An important signaling molecule in this process is cyclic AMP. The structure is shown on page 6. The level of cyclic AMP in the cell is an important regulator of glycogen synthesis and breakdown. Cyclic AMP is produced from ATP in response to rising glucagon levels in the blood. And cyclic AMP can be broken down to AMP, adenosine monophosphate, in response to rising insulin levels in the blood. So, when blood glucose drops, glucagon is produced, and it binds to a receptor on the liver cell and causes cyclic AMP to be produced in the cell. Cyclic AMP stimulates protein phosphorylation, glycogen phosphorylase becomes active, glycogen synthetase becomes inactive, and glycogen is broken down and glucose released. When blood glucose is high, insulin is produced. Insulin binds to a receptor on the liver cell and causes cyclic AMP to be broken down. The enzymes become dephosphorylated. Glycogen synthetase is active, glycogen phosphorylase is inactive, and this results in active glycogen synthesis. Notice that cyclic AMP-dependent kinase, also known as protein kinase A, does not activate the glycogen phosphorylase directly, but it activates another kinase that phosphorylates glycogen phosphorylase. This could be called glycogen phosphorylase kinase. And these are amplification steps. So a small signal can get amplified to a very large response. This is also known as a cascade. There are also phosphatases that remove the phosphate groups, and these can be regulated as well. The phosphatases here are called protein phosphatase 1. Protein phosphatase 1 responds to glucose in the cell. So if glucose enters the liver cell, it immediately activates the phosphatases, and they shut off glycogen breakdown. Okay, now let's look at the regulation of glycogen metabolism in muscle. Glycogen is used as a fuel reserve in muscle. Fatty acids are used as a fuel source until the demand for ATP contraction is high in muscle, and then glucose is used. Muscle glycogen can be used, but only for a short period of time. Muscle glycogen cannot be used to maintain blood glucose levels, or glycemia, since muscles do not have glucose 6-phosphatase. Glycogen metabolism is similar in muscle and liver, but there's some important differences. First, hormone regulation is through epinephrine instead of glucagon. Glucagon is a signal that blood glucose is low and needs to be increased. Epinephrine is a hormone released from the adrenal gland when energy is needed for a quick response. Epinephrine binds receptors on muscle cell and causes an increase in cyclic AMP, which activates glycogen breakdown. Now, insulin has a similar effect on liver and muscle. It activates glycogen synthesis in both tissues. There are some additional intrinsic regulators of glycogen metabolism in muscle. Calcium activates glycogen phosphorylase kinase. One of the subunits of this enzyme is calmodulin. Calmodulin is a calcium binding protein. Calcium is released during muscle contraction, and it binds to calmodulin and activates glycogen phosphorylase kinase. And this stimulates the activation of glycogen phosphorylase and glycogen breakdown, which supplies glucose for ATP production in the muscle tissue. Another regulator is AMP. It can bind and activate glycogen phosphorylase. It actually can bind and activate the dephosphorylated form directly. So when energy levels are low, AMP accumulates, and this stimulates glycogen breakdown for glucose production and ATP production. Glycogen storage diseases are rare disorders that are due to inherited defects in specific enzymes involved in glycogen metabolism. Von Gierke's disease, or glycogen storage disease type 1, affects primarily liver and kidney. The disease is due to a deficiency in the enzyme glucose 6-phosphatase. This enzyme is needed for release of glucose produced either from gluconeogenesis or glycogen breakdown. The disease results in severe fasting hypoglycemia. It causes hepatomegaly due to accumulation of glycogen stores in liver and kidney. The symptoms of this condition typically appear around 3 or 4 months of age. As they get older, children with GSDI have thin arms and legs and short stature. An enlarged liver may give the appearance of a protruding abdomen. Individuals with von Gierke's disease have to have frequent feedings to avoid hypoglycemia. Pompey's disease is a unique glycogen storage disease in that it's due to a defect in a lysosomal enzyme. The enzyme's lysosomal acid glucosidase. Glycogen particles are regularly cleared and resynthesized in cells, and lysosomal acid glucosidase helps clear glycogen particles from the cell. If the enzyme is absent, then glycogen accumulates and causes damage to tissues. Severe cardiac and skeletal muscle myopathy is characteristic of the disease. Quarry's disease is due to a loss in the debranching enzyme, and it affects both liver and muscle. It results in glycogen accumulation in muscle and fasting hypoglycemia. Muscle weakness, cardiomyopathy, and hepatomegaly usually occur. Anderson's disease is due to a defect in the branching enzyme of glycogen synthesis, and it results in very long unbranched glucose chains being stored in glycogen. Now, these long molecules have a low solubility, and it leads to glycogen precipitation and deposits that build up in body tissues. Liver enlargement and damage usually occurs. McArdle's disease is due to a deficiency in muscle glycogen phosphorylase. The primary symptoms are cramping, muscle pain, and myoglobinuria following exercise. And that completes this video on the metabolism of glycogen.